As I was introduced, my name is Ken King, and I work for Ecological Services, more specifically environmental contaminants, at the Lacey, Washington U.S. Fish and Wildlife Office. Previously, I worked as an AIS fish biologist for Lake Michigan out of Green Bay, Wisconsin. And you can tell I'm here to talk to you today about urban stormwater and coho salmon. Before I get started, I want to call attention to our host of partners. They've assisted in providing the funding, expertise, personnel, facilities, equipment, and wildlife needed to investigate this issue. This is a region-wide issue, and it may have not been investigated or investigated as thoroughly without their continued support. I also want to give a special thank you to all the staff that have worked on this issue. These are just some of the individuals that have put long days and nights into multiple projects investigating coho pre-spawn mortality. As you can tell, there's a lot of people you get to know around the area working on this issue. Because many of you may not know the history behind this study, I want to take a brief moment and go over what has led us to investigate the effects of urban runoff on coho. Urban runoff is created when contaminants build up on impervious surfaces and are then rapidly flushed into the nearest waterway when it rains. Here you can see suspended particulates in urban runoff combined with a CSO event flowing into Lake Union. What you can't see are the many thousands of invisible contaminants that are also part of this plume. Stormwater also takes many unseen routes in our waterways. The above video is a time lapse of a submerged stormwater outfall in the Puget Sound during a rain event. Note there's no CSO involved with this at all. For the most part, stormwater isn't front and center for all to see, and therefore isn't a large part of our day-to-day lives. It is, however, a large part of the day-to-day lives of fish in urban creeks. In 2002, the city of Seattle began monitoring adult salmon use of restored urban creeks. As this monitoring continued, they began to notice a pattern of adult coho dying before they could spawn. The average observed coho pre-spawn mortality was a large proportion of the total run over multiple years in urban creeks and a small proportion of the run in rural creeks. The coho suffered from pre-spawn mortality, or PSM. All affected fish exhibited a rapid onset of symptoms, beginning with lethargy and progressing through surface swimming, gaping, loss of equilibrium, pectoral fin splaying, and immobility. These symptoms led to death in a matter of hours. A forensic investigation was conducted to determine where, why, and at what rates returned coho were dying. The phenomenon was documented widely across the Puget Sound area, with mortality rates typically ranging from 60 to 90% of the run. Stressors related to temperature, oxygen, and pathogens were ruled out, and urban runoff seemed to be the cause. After the previous investigation, we were interested in the predicted population scale consequences of the observed PSM. Utilizing life history models, PSM rates were used to predict time to extinction for populations. Local extinction was predicted in 8 to 115 years based on PSM rates of 90 to 20%. However shocking these numbers are, we need to keep in mind they're based off urban runoff having no effect on any non-adult life stage, which may not be the case. We next wanted to see what unmonitored areas may be vulnerable to PSM. To accomplish this, we expanded the sample size from 6 to 55. Utilizing land cover and mortality rates for all sites, we performed a spatial analysis to produce a predictive map of coho PSM for areas around the Puget Sound. The results showed the potential breadth of the issue and that PSM correlated closely with urbanization. With that background, we can jump into the heart of this presentation. Because the likely culprit was runoff, which is known to have components that are harmful to fish at multiple life stages, we created an artificial cocktail of likely suspects to see if we could recreate coho PSM as seen in the field. Water chemistry was monitored from 6 runoff events over 2 years, and representative metals and pHs were mixed at or above levels typically observed, with additional mixtures of metals mixed at 5 and 10 times average amounts also created to account for observed between storm variability. After 24-hour exposures, all runoff did not produce coho PSM symptoms or mortality. Finding that metals and pHs alone were not sufficient to produce symptomology similar to that observed in the field, we started experimenting with the real thing. To answer the question, does runoff cause Coho PSM, we designed an experiment based on exposing adult Coho to runoff in a controlled environment. Runoff was collected from a busy highway in Seattle, which sees an average of 60,000 vehicles a day. Adult Coho were then placed in this runoff or clean well water and exposed for a set time or until symptoms were observed. Runoff was collected and transported within 48 hours to the Suquamish Tribal Hatchery at Grover's Creek to expose returning Coho provided by the tribe. I specifically want to call attention to the tribal hatchery because they donated a significant portion of their Coho to us for many years and all of their Coho to us in some years. For every experiment, 8 Coho were used, 4 in well water and 4 in runoff. To minimize stress, each Coho was placed in its own tube, which were then placed into a tank of 440 liters of either well water or runoff. Fish were exposed for 4 hours during the first 5 experiments and for 24 hours during the last 4. During all exposures, fish were removed and sampled based on symptomology or mortality. During these exposures, conductivity, DO, temp, and pH was monitored and kept within suggested limits. The team found that whereas control fish were active and responsive, as shown in the video on the left, the runoff exposed fish in the video on the right showed the classic suite of PSM symptoms. In total, we exposed Coho to runoff from 9 rain events over 3 years. 100% of the control fish survived, while 100% of the exposed fish were either symptomatic or dead during the same time frame. These experiments demonstrated that 100% urban runoff consistently recreated Coho PSM symptomology or mortality similar to that seen in the field. With urban runoff identified as containing the source of Coho PSM, we wanted to find potential ways to mitigate its effects. Because the issue was caused by runoff and increases with urbanization, we started looking at green stormwater infrastructure to see what effect it could have on Coho survival. Green stormwater infrastructure are technologies designed to help runoff water infiltrate into soils. The basic idea of GSI is to take stormwater and slow it down, spread it out, or soak it up. We chose to focus on inexpensive, non-proprietary, transportable GSI options that are easy to implement, both public and privately. This set of requirements led us to bioretention. 4 bioretention cells were assembled using 55-gallon drums that could be moved on a single pallet. These drums were constructed according to the Washington Department of Ecology specifications, utilizing mulch, compost, sand, and gravel. To test the effectiveness of our cells, we filtered runoff through them at a consistent rate and exposed Coho to either filtered runoff, unfiltered runoff, or well water. Coho were exposed to the treatments utilizing the same experimental equipments, methods, and monitoring as in the previous experiment. The filtration experiment was repeated 5 times over 2 spawning years, and the results are shown in the table above. The first exposure was for 4 hours. At this point, half the fish in the urban runoff were sick and half were dead. The team was concerned that filtered and unfiltered runoff may have the same effects but act on a different time scale. So, for the final experiments, the exposure time was stretched to 24 hours. At that point, the results were quite stark, 100% dead and unfiltered, with 100% live and non-symptomatic in both filtered and control water. Here you would have been able to see the video results from that experiment. I will briefly describe it to you through the security measures on the computer. These top 2, you see fish happily swimming around, quite active, and we give them a very light touch, and they immediately move and seek cover around this observation tank. However, on the bottom one, the fish is extremely non-responsive. It's losing its equilibrium and gaping. In conclusion, we could not recreate Coho PSM artificially using metals and PAHs. We could, however, cause Coho PSM symptoms and mortality by exposing the fish to urban runoff. Additionally, all symptomology and mortality was eliminated at all timescales tested by filtering runoff through bioretention cells. This demonstrated that highway runoff contains the contaminants both necessary and sufficient to cause Coho PSM. That concludes what I came here to talk to you about, but from previous presentations, we have learned that stopping here leaves many unanswered questions. With that in mind, I'll briskly go over additional results that have occurred since this paper was published. There are many different experiments on a variety of species, as well as juvenile coho, looking at the effects of bioretention on abnormalities or mortality caused by runoff. These experiments, summarized in this table, show that bioretention can reduce or eliminate the effects from urban runoff on all species tested. Beginning in the late 2015, we developed coho embryos in 4 heath stacks over a total of 64 days. During this time, each stack was exposed for 48 hours to 15 runoff events. Exposures consist of either 100% runoff, 50% runoff, filtered 100% runoff, or well water. At the end of the experiment, we saw a significant decrease in their survival of embryos treated with 50% and 100% runoff as compared to the controls and filter treatments. In addition, when the 100% runoff was filtered through our bioretention cells, there was no residual effect on survival. To investigate the effects of urban runoff on other salmon species, returning adult chum and coho were exposed to either runoff or control water for 4 hours. The adult chum on the left, you can see the rippling water was actually splashing water out of the observation tank after 4 hours of stormwater, whereas the coho on the right, after 2 hours, is not even gilling in regular intervals and is just about to call it quits before we took its samples. And that was fairly consistent. The chum has exhibited no symptoms past lethargy over all the experiments. We repeated it 5 times over 1 spawning season. During the winter of 2015, we exposed spawning age hatchery coho that had never transitioned to saltwater to urban runoff for 6 hours to see if they would exhibit PSM symptoms similar to those seen in saltwater return coho. This experiment was repeated 3 times, and although the sample size was small, we were able to elicit coho PSM symptoms and mortality similar to that seen in the field. In a continuing effort to track down the root cause of coho PSM, tissues and organ samples were taken from fish beginning in 2012. In 2015, multiple blood samples were taken from all fish to better understand what is happening inside symptomatic fish. With this data, we are compiling a unique symptom profile for coho PSM, which can assist us in identifying runoff-related mortality and in narrowing down the potential causal agents. Are there any questions? So you're asking how they could work on a larger scale in urban settings. Potentially, they could be scaled up, but also a lot of the downspouts, I don't have a photo of my presentation, are small pipes, maybe 4 to 8 inches in diameter. They have runoff coming, but there's hundreds of them along highways. So potentially, we're also investigating what's happening inside these cells and to see, mainly because we don't want the cells to become just a tiny Superfund site at some point in the future. But we're also seeing if they can process the amount of runoff needed. And actually at the NOAA Sandpoint facility, they're one of our partners, they're redoing all their entire stormwater runoff for a few hundred acres and trying to treat it as kind of a pilot program using the research that we've been doing. So it's starting small, looking at it. And then this is the sort of thing that you could potentially put in your house if you could treat your own stormwater runoff, which may be coming on some of these small urban creeks. But as far as large scale, that's kind of beyond what we're looking at with this.
Do you investigate that at all?
We've tested a lot of the stormwater. It's a little bit hard to break it down since there's potentially thousands to hundreds of thousands of chemicals, and it's a moving target. A storm is not a storm and a fish is not a fish, but we do see these large trends. We initially thought like you did and tried to break it down using the known bad actors, but once we started doing some more testing, looking at everything that was in it, we kind of gave up on that PAH because it wasn't eliciting the same fast response death that we saw in the fish, and moved on to actually building up. We were analyzing blood chemistry to seeing what's happening inside the fish, and then using that basically to cut out, it's probably not something that can affect this in the fish. We have some preliminary ideas about what's going on that we need to do experiments in these coming years to solidify what's actually happening inside the fish. And then from that point, we're going to be looking at what potential actors could be causing it. Does that answer your question?
Yes, thank you.
Do you have a question now?
I do. Two quick questions for you. One is, so the filtration is encouraging. I'm wondering if you know, at some point, do the filters essentially get filled up and they no longer work? First question. Second question is, for the storm events, how quickly after the storm event do you no longer see the effect on the fish?
Very good questions. We're currently investigating a little bit more in these bioretention cells. There's been preliminary work from somebody in Australia that has been having used them for 10 years, and they're still not toxic, still filtering, not for this particular Coho mortality, of course, but as just generically filters. It's something that definitely needs to be addressed before this becomes implemented on a large scale. And then, could you repeat the second question one more time?
Yeah, basically, I was wondering how quickly after the storm event you didn't see the effect on the Coho.
Okay. We're doing juvenile studies. Currently, we're about to start in piloting to kind of get at that at what dilution and at what time scale does it affect them. Currently, it comes through as a pulse, and it's just dead fish everywhere. So there's really not that much of a time scale from the surveys we have done that it seemed to be. Usually we chose at 4 hours based on field observations, but a lot of them would go down even in 1 to 2 hours, just complete mortality. So it's hard to say for sure based on the stream surveys and field observations.
So my quick question is, with the treated water fish, did you look at sublethal effects? Like, was there reduced fecundity in those adults? Like fewer eggs compared to the control fish?
We've been beginning to look at sublethal effects. We're still analyzing that embryo study that I showed, the mortality, but the sublethal effects of the exposures to duration. And we're kind of getting the big picture items because we're using also 100% stormwater, which isn't necessarily representative of what they're seeing in the field. We did do some effects using actual urban stormwater from streams that saw high death rates, and that showed a host of sublethal effect in developing embryos and juveniles. Although we can't contribute that all to urban runoff. That's a big caveat with that. We just pipe what was in the stream into developing tanks.